A pleasant day to all. I am Joyce G. Nilo.
I am Alexis Lagac.
I am Abigail B. Sanchez.
I am Jamia G. Aquino.
I am Philippe Lacsamana.
Presenting our research entitled Anti-angiogenic Activity of Santan Leaves Ethanolic Extract using Philippine duck egg embryo Chorioallantoic membrane assay.
Angiogenesis is a process of forming new blood vessels from pre-existing vasculature, Carmilla 2003. It is a crucial process for a number of physiological processes, including the growth, healing, and function of a female reproductive system, Assay and Tehergora V 2012. However, according to Musa and Rahabi 2017, anti-angiogenesis takes a vital part in the progression of tumors. The process of tumor anti-angiogenesis is crucial to the survival of solid neoplasm, Casanova and Gasselu 2019. In view of this, scientists have developed drugs called anti-angiogenesis inhibitors that block tumor anti-angiogenesis, which hinders or decelerates the growth of cancer, National Cancer Institute 2018. However, there have been reports of high risk of side effects, albeit approved by the Food and the Drug Administration, Saladis-Kalman 2022. Thus, there have some medicinal plants that have been studied and discovered to be used as an alternative for the lower risk of side effects. In view of this, the researchers discovered santan, which is a plant found to yield a specific type of flavonoids such as CAM, quercetin, lutein, and protein. AJ et al. 2018. These specified flavonoids have already established its profile as an anti-angiogenesis agent. Araujo et al. 2011, Chen et al. 2019, Cheng et al. 2021, Barbosa et al. 2017.
In general, this study aims to determine the anti-angiogenic activity of santan leaves ethanolic extract using Philippine duck egg embryo CAM Assay. Specifically, this study aims to answer the following questions. What is the anti-angiogenic activity of santan leaves ethanolic extract at the following concentrations: 25%, 50%, 75%, 100%, and negative control? What is the minimum and maximum anti-angiogenic activity concentration of santan leaves ethanolic extract? Is there a significant difference between the anti-angiogenic activity of santan leaves ethanolic extract at the following concentrations? The study was centered on a true experimental research design. This design focuses on investigating the variable's relationship by controlling an independent variable to evaluate its effect on dependent variables. As shown in the table, the researchers have categorized the following variables evident in the study. Proceeding to the sampling, 2 kilograms of santan leaves were collected, while 61-day-old Philippine duck eggs were purchased by the researchers. Furthermore, an image of the plant's flower, leaves, and stem was sent to Far Eastern University Herbarium for authentication. In carrying out the experimental procedures, the researchers have utilized the following materials and equipment. For the actual experimentation, the first phase was the ethanolic extraction of santan. The 2 kg of santan leaves was air-dried at room temperature for 3 days, and with the aid of the household blender, the air-dried leaves was pulverized. The powder of the leaves was soaked in a 95% ethanol for 24 hours. After soaking, the solution was filtered and subsequently put in the rotary evaporator and went through dilution with distilled water. In the second phase, an in ovo Chorioallantoic membrane assay was performed. After cleaning the fertilized eggs, it was placed inside the incubator at 37 degrees Celsius for 72 hours. A needle was used to drill through the air sac for air displacement, then a small window was made through the use of tweezers to allow a visible area of blood vessels. A clean cling wrap was used to seal the eggs before it was put back in a static egg incubator for the same temperature. After 3 days, the diluted extracts were placed in a petri dish where the filter paper disc was soaked. Subsequently, the disc was administered onto the CAM and incubated once again at 37 degrees Celsius. 5 treatment groups were developed, and the table here exhibits the dilution computation of the following concentration. For the data gathering procedure, through the use of stereo microscope and camera, Analysis and quantification of the blood vessels was performed, in which the imagery software enhanced the images to improve the visibility of the blood vessels by adjusting the saturation, brightness, and contrast. Proceeding with the manual tallying of the first blood vessels or smallest branch points, the quarter of the image was only considered. On the other hand, the data was analyzed by employing descriptive statistics, such as one-way analysis of variance for obtaining the difference between groups of concentrations, and TACI-HSD post-hoc analysis for multiple comparisons on the effect of each treatment.
Proceeding to the results and discussion, in Table 2, the study found that Santan leaves ethanolic extract at different concentration can inhibit blood vessel growth at 25%, 50%, 75%, and 100%. Each formed 25.5, 26, 25, and 23.5 blood vessels. The negative control developed an average of 47.5. This suggests that increasing the concentration of the extract and suppress blood vessels growth. It is in line with Castor et al. 2014 observation in conducted study in which the flavonoid, specifically quercetin, a phytochemical component that coccinea also inhibits the growth of blood vessels at its concentration increases. The table above utilized a Tukey post-hoc test which shows the p-value that each group of concentration obtained when compared to one another. The group of 25%, 50%, 75%, and 100% concentration gained a p-value of p is greater than 0.05, excluding the comparison with the negative control, indicating no significant difference, since p is greater than 0.05, thus failing to reject null hypothesis. On the other hand, the negative control group has acquired a p-value of p is less than 0.05. It reveals that the four concentration groups of Santan leaves ethanolic extract, specifically the 25%, 50%, 79%, and 100% showed a highly significant difference when compared with the negative control. Since p is less than 0.05, then the null hypothesis rejected. The results agreed with the study of Salas and Tataan 2015 wherein the extract of sabongay, pandan, and siangkubat containing similar phytochemical components with santan exhibited significant difference in the average number of blood vessel branch points formed by the plant extract when compared to negative control. With the increase of concentration, a significant decrease is observed in the number of blood vessel branches. The table exhibits a statistical analysis using a one-way analysis of variance, in which the p-value obtained between groups of concentration is p less than 0.05. It exhibited there is a highly significant difference between the anti-angiogenic activity of Santan leaves ethanolic extract at concentration 25%, 50%, 79%, 100%, and negative control. Since p is less than 0.001, the null hypothesis is rejected.
Findings: increase in concentration of the extract leads to occurrence of anti-angiogenic activity as supported by the conducted study of Magdadaro and others 2023. The anti-angiogenic activity may be due to the presence of phytochemical components such as alkaloids, flavonoids, glycosides, saponins, and tannins. Acquired from the statistical analysis, it additionally implies that as the Santan leaves ethanolic extract concentration increases, the ability of the extracts to suppress the growth of blood vessels also increases, corresponding with the study of Mia and others 2010, whereby the increase of concentration of spinosentrum gallianum extract is proportional to the increase of angiogenesis inhibition activity, albeit a small difference. While between the treatment groups and negative control, there exists a highly significant difference. In addition, a 25 concentration of the extract is enough to target blood vessels proliferations, similar to the study of Zihlif and others 2013. This study concludes that Santan coccinea leaves is a possible source of agent targeting cancer through inhibition of angiogenesis.
For the recommendations, thus the researchers recommend to conduct further investigations regarding this plant due to their potential as an anti-angiogenic agent. Screen other parts of the plant that have similar phytochemical components that target angiogenesis. Utilize other solvents for extraction that can increase the efficacy of the anti-angiogenic properties that the plants possess. Perform histopathologic examination to assess possible undesired effects that the extract may cause. Allot a greater time frame for the conduct of the experiment to attain more effective results. Inclusion of other conditions in determining the anti-angiogenic activity.
This is where our presentation ends. Thank you.